ARCS is a national organization that began in Los Angeles in 1958 by a group of visionary women who were determined to elevate science in America by creating a unique partnership between academic science and society. Since then, ARCS has grown to 15 chapters and donated over $100 million to over 10,000 ARCS scholars across the country, with 100% of funds raised for scholar awards actually going to the scholars.
I'd like to thank the ARCS organization for their generous support of my research.
I wanted to thank everybody at ARCS and everybody who's supporting the scholars and this wonderful program. Now that I'm finished with my Ph.D., I plan on supporting this program as well.
ARCS Foundation Orange County identifies brilliant young American scientists and encourages their achievements. This is your opportunity to be directly involved right here in Orange County by helping ARCS to support the bright young scholars at the University of California, Irvine.
The significance of ARCS has been understated locally and nationally over the years. It is not known nor appreciated that science and engineering faculties have to raise competitively the funds they need to support their graduate students financially, to create, equip, and maintain their research laboratories. The ARCS scholarships are basic financial support, not only to the students, but also the faculty.
I encourage you to become part of the ARCS community and make a personal difference in the advancement of science in Orange County. Enjoy meaningful friendships and be inspired by the positive potential of our scholars' sophisticated research that you personally support. We embrace and welcome you to become a member of ARCS OC and a supporter of college scholars on the front line of scientific advancement in America.
I'd like to thank the ARCS Foundation for your support. With my ARCS funding, I've been able to get a new laptop, new research books and software, and support my research while working from home.
The funding from the ARCS Fellowship actually has enabled me to attend multiple conferences where I've been able to present this work. So I really can't thank ARCS enough for their support.
I just wanted to say a big word of thanks to you guys for continuing to support us in our scientific research during this very unusual time.
Due to your support, I was able to share my work at multiple international conferences, and I was able to have multiple resources to thrive in grad school.
ARCS has also provided me with the support that I need to purchase electronic materials to analyze data, software to analyze data, as well as travel expenses for collaborations and also for conferences.
Thank you again for supporting me and allowing my research program to thrive.
I invite you to join our family of prestigious philanthropists, foundations, and corporations by giving to ARCS Foundation Orange County Chapter. We support 20 scholars a year who are in need of your help. This recognition not only validates your research, but also imbues them with a new confidence that comes from knowing that their work is meaningful, important, and extraordinary.
To give, click the donate button on our website. On behalf of all the ARCS Orange County members and the scholars, we thank you.
This year, due to the special circumstances we live in, we are proud to present our 2020 Scholars Research in this virtual format. Please enjoy these presentations and make a donation to support a new scholar to ensure a bright future for advancing science in America.
When you get sick, you need your immune system to help you feel better. That's what happens when you get a cut or when you get a virus. But what about in the brain? In diseases of memory loss, like Alzheimer's disease, which is the most common form of progressive dementia, people knew that the immune system would respond to this disease, but they didn't really understand that this response can actually modulate whether or not someone gets Alzheimer's disease, until recent genetic studies were able to show that the mutations associated with increasing risk of this disease affect the immune system. The problem now is that we don't really know what they're doing. Do people with Alzheimer's disease have a hyperreactive immune system, or is their immune system too suppressed? So in order to figure this out, I'm studying 2 of these new Alzheimer's disease mutations. One associated with increased risk for Alzheimer's disease, and a protective mutation associated with decreased risk for Alzheimer's disease. What I've found is that the mutations that are increasing risk for Alzheimer's disease make it so that the immune system is not responding to the disease. And likewise, the protective mutations, or someone's less likely to get Alzheimer's disease, are actually making the cells better at responding and clearing the disease. So this is really exciting data because it shows what we need to do in creating a vision for new drugs and new therapeutics against Alzheimer's disease. We need to be able to reactivate the immune system so that things are acting more like these protective mutations and giving the immune system a chance to fight back against Alzheimer's disease. Thank you.
Hello everyone, I hope you're all staying sane and healthy. I'm sorry that we couldn't be together for our normal ARCS dinner where I could have presented this work to you in person, but I'm excited to still get the opportunity to share with you the work that I've been doing during my Ph.D. program in the lab of Ziva Wunderlich, working to understand how different genetic systems are able to achieve normal embryonic development. And so with that, I'm going to go ahead and share with you my presentation so you can see that. So when we think of genetics and DNA, we often think of genes that encode a protein that performs some function. But the large majority of our genome, 99% of our DNA, actually does not encode proteins. And so when we first realized this, there was kind of this question in the field, why would so much of our DNA consist of this junk DNA? But biology is quite clever, and over the past couple of decades, it's become clear that this non-coding DNA is anything but junk. It often plays very critical roles. Much of this non-coding DNA are regions that actually control the activity of protein-coding genes. One critical class of this regulatory DNA are enhancers. And so enhancers are sequences of DNA that control the activity of target genes. And enhancers work by binding key effector molecules called transcription factors that I'm showing here as the different colored circles. And when the right transcription factors are available, an enhancer will turn on its target gene at a particular time or place. And so that's what we're looking at here. We're looking at a developing mouse embryo where gene expression is being controlled by an enhancer, and that's what we're looking at in blue, where gene expression is being told to be turned on in the spinal cord by a particular enhancer. And so by controlling gene expression in this manner, enhancers play critical roles in development. It is through regulation by enhancers that, for example, a muscle cell knows to become a muscle cell, while a nerve cell knows to become a nerve cell. We can see just how important enhancers and other non-coding regions of DNA are simply by the fact that 93% of the disease-related mutations that we know about occur in these non-coding regions of the genome. And so my project focuses on understanding a particular kind of enhancer that seems to be quite pervasive in developmental systems. These are called shadow enhancers. Shadow enhancers are groups of multiple enhancers that all control the same target gene and turn on that target gene in a very similar pattern. And these shadow enhancers have been shown to be critical for proper development under conditions of stress, but no one knows exactly how or why how these shadow enhancers are able to function. But a key characteristic of shadow enhancers that I hypothesize is critical for their function and development is that the individual member enhancers of these shadow enhancers groups bind different transcription factor, those effector molecules, that allow them to activate their target gene. So that's where we're looking at this example here, where our one enhancer called proximal is activated by these transcription factor molecules, while the second enhancer, the distal enhancer, is activated by these two different transcription factor molecules. Yet both of these enhancers are able to drive a similar pattern of expression of their target gene. And we think it is through that this, what we're calling a separation of inputs, that shadow enhancers are able to buffer against fluctuations in the levels of these transcription factor molecules, and therefore drive more stable levels of expression of their target gene. And so in order to actually test this hypothesis and assess how shadow enhancers are functioning to ensure robust development, I visualized the activity of these enhancers in living Drosophila embryos using fluorescent microscopy. And that's kind of the pipeline we're looking at here. And this is an example of some of my imaging runs. And by tracking the activity of these different enhancers across developmental time, I can learn about how they each function to control gene expression. And so with help from my ARCS funding, I've made significant progress on this project. I actually have a paper that we're currently in the process of resubmitting that shows indeed, kind of circled here, a shadow enhancer pair drives more stable gene expression than single enhancers or duplicated enhancers. And this is true even across a wide range of temperatures. So this is really exciting finding that was a really nice conclusion to this project. And so my findings may underlie why exactly shadow enhancers are so common in developmental systems. And these findings might help us better understand the mechanisms by which development not only functions normally, but also why it can sometimes go awry, and hopefully inform future efforts to prevent some of these developmental issues. And so with that, I'd like to thank everyone in the Wonderlic Lab for all of their help and support, as well as a big thank you to our collaborators in this project, as well as obviously a huge thank you to ARCS for all of their help and support during my time in my PhD program. The funding from ARCS, the ARCS Fellowship actually has enabled me to attend multiple conferences where I've been able to present this work. So I really can't thank ARCS enough for their support. And I thank you all for listening. And again, I hope you're all staying sane and safe, and I hope to see you all again in person very soon.
Hello everyone, my name is Mariam Asghari and I'm a 5th year PhD student at University of California, Irvine in Mechanical Engineering Department. Today I want to talk about my research, which is using solid oxide fuel cell technology, integrating with this technology with liquid desiccant to provide cooling and power for data center application. Just think about how much you are dependent to the internet technology. How many emails do you send per day? Or now that you are at home because of COVID-19, how many Zoom meetings do you have per day? All of these are dependent to data centers. Right now, data centers are responsible for 2% of the electricity in the United States. But in 5 years, they are going to be responsible for up to 20% of the electricity in the United States. Right now, data centers are responsible for 2% of the world's carbon emission, but in 5 years, they are going to be responsible for up to 6% of these carbon emissions. If you look into traditional data centers, only 17% of the energy of the fuel goes to the server, which is very inefficient. And if you look into data centers, there are two big consumers of energy. The first one is servers, which is exactly where we want our energy to go. But the second ones are cooling systems. Data centers need cold and humidified air to prevent any damages to servers. As the society relies more and more on internet technology and data centers, there are needs for more reliable data centers. Right now, data centers need 99.49% electricity reliability, but electrical grid can only provide 99%. Only in 2017, there have been more than 3,500 power outages due to natural disasters. And for every single second of downtime of data centers, eBay data center, they have lost $6,000. And for all of these downtimes, we are losing our data. So the solution for an efficient and reliable data center is using solid oxide fuel cell technology, which is fuel flexible, has high efficiency and low emission. And that's exactly why big companies like Apple, eBay, and Equix, they have been using large-scale fuel cells for powering their data centers. A couple of years ago, National Fuel Cell Research Center, in collaboration with Microsoft, they investigated the concept of using one small-scale fuel cell for powering one single server rack, which eliminates the need for backup generator and also eliminates all the inefficiency associated with distribution and transmission lines. But using these high temperature fuel cells gives us the opportunity for cogeneration application because of the high quality exhaust that is available. And that's why I introduced the idea of not only using one small scale fuel cell for powering one single server rack, but using the exhaust of this single fuel cell for producing cold and humidified air in a desiccant humidifier to provide the cooling that we need for one single server rack. I investigated this idea both theoretically and experimentally. I used my model to look at 7 different locations that Microsoft has data center in the United States. 3 of these locations need both humidification and cooling. So my results shows that if we run our side oxide fuel cell technology continuously and provide cooling and produce cooling, we are able to provide between 15% to 25% of the cold and humidified air that we need for one single server rack. There is also another scenario that we can look at to the storage, which means that, for example, for a place like Virginia, which only needs dehumidified air only 6 months between May and October, we can use the rest of the year to produce a strong desiccant and store this strong desiccant in a storage tank. And then when we need dehumidified air, we use this strong desiccant to produce cold and dehumidified air for our data centers. And when we look at this specific scenario, we are able to provide not only the power that we need for our single server rack, but also we are able to provide the cold and humidified air that we need for that one single server rack. Thank you for your attention, and please email me if you have any question.
Hi, my name is Chloe Groom, and I'm a 2019 ARCS scholar. My research uses quantum chemistry to find ways to make hydrogen fuel cell vehicles more affordable. Nobody likes breathing smog. If you can afford it, driving a Tesla is a great option to drive greener. My research advisor owns one, and I won't lie, I'm a little jealous. Even so, we do research together on another type of green car that runs on hydrogen gas. Better than Teslas, they charge faster and can go farther on a single charge. But just like Teslas, they're also expensive. And this is because the batteries in hydrogen fuel cell vehicles need platinum metal to work. And like any rapper can tell you, platinum is better than gold. Unfortunately, the price tag reflects that. A single handful of platinum is more than $5,000. So for hydrogen cars to be affordable, we have to find a way to replace the platinum. We know that single atoms of iron, stuck to special surfaces, come close to the performance of platinum. As gas flows through the cell, it lands on these single atoms and they get broken apart, which drives the chemistry. Unfortunately, even single atoms of iron aren't good enough. We can't go smaller than a single atom, so I decided to explore other metals besides iron. Luckily, I do computer simulations, so I can look at more metals much faster than running experiments. The problem is that even though I found other candidates that were better than iron, they still couldn't beat platinum. I thought about literally simulating every single transition metal on the periodic table, but I didn't want to spend months in front of my computer screen ripping my hair out. Then I realized that I'm forgetting something. The surface. These single atoms, as I said, are stuck on a surface. By changing the chemistry of the surface, I actually made the metal atoms magnetic. I'm still working on understanding why, but making them magnetic increased their performance by over 40%. I think it's because the additional magnetism helps tug the gas molecules apart that land on the single metal atoms. Going forward, I'm doing more simulations, looking at how the magnetism changes with the surface chemistry. I'm this close to beating platinum. This has huge implications for the future. Without platinum driving up the cost, we could all be driving hydrogen cars that are much better for the planet. We can be better than a Tesla without the price tag. Thank you so much.
Hi everyone, my name is ELISA Thone, and I'm a 5th year Ph.D. student in chemical and biomolecular engineering, and I'm excited to share a little bit about my research with you today. When it comes to taking on cancer, we hear a lot about early screening, better detection, and advances in therapy. But what about prevention? Getting more exercise, avoiding smoking, and even articles suggesting diet changes, such as cutting out coffee, can help with prevention. But none of these are a sure bet. And I don't know about you, but I'm definitely not giving up on coffee. So what else can we do? The answer right now is not too much. But that is an issue I've committed to changing. In my research, I'm working to create a preventative vaccine so that one day we have a proactive approach to preventing cancer and we can take action before the patient ever gets sick. So how do vaccines work? Pieces of the dangerous substance you want to educate the immune system against are injected into the patient with the hope that they will be taken up by the antigen-presenting cells and presented on their surface to T cells and B cells to educate them on what to look for, with the end goal of being to produce memory cells that will always remember to be on the lookout for that dangerous substance. Traditional approaches have included injecting cancer proteins or nanoparticles, but were rapidly cleared from the body and were not effective in preventing tumor growth. So somebody thought, why don't we skip the middleman and just use antigen-presenting cells as the cancer vaccine itself? A company called Dendreon did exactly this when they made Provenge, which is a dendritic cell cancer vaccine for prostate cancer. This was approved by the FDA in 2010 because during clinical trials, it showed to be safe and improved survival. However, less than 1% of the dendritic cells reached the lymph nodes. That's where they need to go to educate the other immune cells. And control of maturation state was also an issue. To me, that's the most important part because an immature dendritic cell requests a tolerance response, while a mature dendritic cell will demand an attack. And in the case of cancer, that can be the difference between life and death. Ultimately, in 2014, Dendreon filed for bankruptcy. And while we think they were on the right track, the ideal platform would maintain the cell-to-cell communication ability of these dendritic cells, but with more control. And we think that extracellular blebs, or EBs, might be a promising solution. Extracellular blebs are particles made from cells in the micro and the nano scale. Up close, they're phospholipid bilayers, identical to cells, with all molecules needed for cell-to-cell communication built in. Unlike cells, they're not living, and once produced, they can't change or lose efficacy. In other words, you gain a lot of control. So what we're doing throughout this work is isolating bone marrow from our patients, differentiating that bone marrow into immature or mature dendritic cells, and then making particles from these, the extracellular blebs. And so throughout this, we're testing these 4 groups: immature cells, mature cells, immature particles, and mature particles in their ability to activate T cells to eradicate tumors. So in order for these particles to work and stimulate immune cells, they have to look identical to the cells they are produced from. So we investigated how similar they were. We took immature cells and incubated them with a cancer peptide and a chemical that induces maturation over time. At various time points, we checked how similar the EBs were to the cells, and we found that they were nearly identical. At this point, we knew the EBs could talk the talk, but next was to test if they could walk the walk. In other words, would they actually work? In order to do this, we performed a tumor challenge where we vaccinated mice twice and then administered cancer cells expressing the protein, which, in theory, the immune system should have been educated about. You can see pictures from our study of day 30 here, with our controls, saline, cancer peptide, and cancer protein, all showing large tumors, as these have never worked as effective cancer vaccines in the past. Our mature cell and particle groups here show almost no tumors at day 30. And what's the most interesting, in my opinion, is the immature group. So again, this should be the group of asking for a tolerance response. In the cell vaccine group, you can see almost no tumors, while in the bleb or particle group, you see a large tumor. If this was truly a tolerance response, both should look the same, but it's possible that the cells, because they're living, changed and requested a different response. So this goes back to that ability of having higher control with our particles. If you look at the data more long-term, so this graph on the left, we have tumor volume on the y-axis and time on the x. If we focus on the red and the purple data, which is the mature cell and particle groups, you can see that over time, tumors were totally eradicated. And if you look on the graph on the right-hand side, this resulted in 100% survival rates. So in conclusion, we developed a cell-free, highly controllable cancer vaccine. The blebs, or these particles, mirrored the parent cell's molecular presentation nearly identically, and they were capable of preventing tumor growth and eradicating tumors in vivo. I'd like to thank NSF GRFP, UCI Graduate Division, and of course ARCS for all of your funding. Thank you, it's meant a lot and it's done a lot for me. Also my lab mates for their contributions. And if you have any questions, please contact me at my email below. Thank you.
Hello, my name is Bryce Wilson. I'm a first-year scholar working under Professor Venugopalan and Professor Botvinnik in the Chemical Engineering Department. My research is focused on exploring how physical forces like touch, strain, and flow play a role in cell biology.
I want you to imagine a game of telephone. In a game of telephone, you whisper a message into somebody's ear and it gets passed down the room. The fun of the game of telephone is to see how much the message has changed by the time it gets to the end of the room. Biology is the language of life, and it's spoken in these games of telephone. And as a biologist, what we want to do is precisely understand this game given any room. So how we can do this is to basically mic up everybody in the room. So given any message, how is that person likely to pass it to their neighbor? And we can think of a disease as when this game of telephone goes wrong. I don't mean somebody changing the message, that's the whole point of the game. What I mean is the message being completely lost or changed in an incomprehensible way. Now I'm going to change this game up a little bit more. In this game of telephone, instead of whispering in your neighbor's ear, what if half of the students in the room wrote the message on each other's hands with their finger? Now the microphones don't work anymore. We could possibly lose this message somewhere in the row, and we don't understand how it's being changed or how it's being lost. So when cells are communicating with touch and passing these signals down biochemical pathways, our existing probes or our microphones don't work anymore. What my team does is we've developed the first probe to figure out how we can study cells when they're communicating with touch. So what our probe is basically a bubble. We focus a laser down nearby the cells, and this bubble expands out. And when the bubble expands out, a tiny tsunami flows away from the bubble. And the cells feel this tsunami in the same way they'd feel blood flow, for example, or trauma. There's some advantages to our technique. First of all, it's variable in force. We can create a large bubble that kills a bunch of cells and is a huge force on cells that would mimic, for example, a traumatic brain injury. Or we could create a small bubble that induces a flow very similar to the blood flow in cells, and we can study heart disease with it. This is a very scalable renovation, and it can be used in a 96 well plate and test hundreds of drugs at a time. In the future, we hope to expand this technology into 3D spaces. None of the cells in your body exist on a plate in a 2D sheet. All the cells in your body exist in a 3D tissue. So in the future, we hope to find ways to expand this bubble into tissue, which would be analogous to a balloon expanding. This is very different than a wave of blood flowing over the cells. So I want to talk about traumatic brain injury again. When we think of traumatic brain injury, we're thinking of an NFL player sustaining many concussions over his career. Well, it turns out the line in the sand isn't at concussion anymore. We're seeing symptoms of traumatic brain injury with people that play soccer, for example, from heading the ball. So if the line isn't at concussion anymore, where is it? We thought of an experiment where we can induce bubbles of larger and larger sizes to try to find out exactly when this disease condition occurs. Now, this is only one experiment. There are other experiments, like with heart disease, for example. We can induce higher and faster flows past these cells to try to see when heart disease conditions start to occur. We think this can really change the field of mechanotransduction, and we're really excited.
We all want to know the future. I want to know if I'm going to have a heart attack or if my mom will develop symptoms of Alzheimer's disease. Predictive models allow us to answer a lot of these questions. However, predictive models are only as good as the data we use to train them. This is a problem in lots of different scientific disciplines. For example, predictive models are used in the criminal justice system to predict who is likely to commit a second offense. In 2016, ProPublica published a paper looking to see how accurate these models actually are. And they looked at the individuals who are labeled as high risk for committing a second offense. Now, they first looked at white individuals who are labeled high risk, and they found that 75% of them actually went on to commit a second offense. So the model seems to be working pretty well, and it's pretty accurate. However, they looked at the African American individuals who are labeled high risk and found that only 45% of them went on to commit a second offense. Less than half. So this model isn't accurate, and it's less accurate for the African Americans. This happens because the data used to train the model overrepresents white individuals. So the model actually learns to do a better job at making predictions for the white individuals. In order to solve this problem, we either need to get data that's more representative, or we need to measure the differences between the data used for training and the actual population in the US that we want to learn about. This example is a perfect analogy for problems we're facing in medical research today. My research is focused on predicting who will develop symptoms of Alzheimer's disease. And we want to do this using a predictive model. And we want to do this with clinical trial data because we have a lot of it from all the clinical trials we've run. However, people have to volunteer to be a part of clinical trials. And it's been shown that clinical trials tend to overrepresent white individuals. And so with current predictive models, we would get the most accurate predictions for white individuals. Again, just like we got the most accurate predictions for white individuals in the criminal justice example I gave you earlier. My research is focused on measuring the differences between the clinical trial participants that we have and the population of all elderly adults in the United States that we want to learn about, and accounting for these differences. This will allow us to obtain more accurate predictions so that when you or I go to the doctor, we can trust the predictions that we receive. Thank you.
Hello, ARCS Foundation members and fellow ARCS scholars. My name is Amanda Cullen, Ph.D. candidate in the Department of Informatics in the Donald Bren School of Information and Computer Science. This presentation, Gendered Stereotypes, Harassment, and the Work of Video Game Live Streaming, focuses on an aspect of my dissertation examining the experiences of women who work as video game live streamers in order to understand differences in labor, personal expression, and sociality in online gaming contexts. The online gaming context I focus on in this presentation is the live streaming platform Twitch.tv. Twitch is a site like YouTube that is filled with user-generated content, but the focus on Twitch is live performance with real-time communication between a streamer and a set of viewers. There are over 3 million streamers on Twitch, and the platform has an average of 15 million daily visitors. The content of channels on the site, like those seen here, are largely video game focused, and many of the social norms and behaviors on the site are derived from those found in video game culture. Live streaming is an explosive sector of technology and communications industry growth, as well as a prominent facet of the billion-dollar video game industry. Video game live streaming is also becoming one of the most influential ways that technology users interact with digital media and online communities. And I'll also add that the events of the past few months have highlighted the importance of online sociality infrastructure and live streaming platforms. Live streaming offers valuable opportunities for players and viewers to express themselves, form interpersonal connections, and develop social and technical skills. However, on Twitch, the ubiquity of norms and attitudes prevalent in video game culture, like meritocracy and misogyny, make it difficult for women to take advantage of opportunities in the same way. In fact, I argue that heightened opportunities for success and visibility offered by live streaming platforms, as well as their reproduction of values inherent in video game culture, create an environment where the participation of women is often evaluated according to gendered stereotypes. My dissertation is an ongoing digital ethnography using multiple methods such as surveys, discourse analysis, participant observation, and interviews. This combination of methods allows me to observe behavior and communication in natural settings, to question participants directly, and to highlight issues in the cultural contexts around live streaming. Through over 100 hours of participant observation on live streams and an analysis of dozens of threads in streamer forums like this one, I have documented many examples of women encountering sexist remarks and rhetoric while performing the work of live streaming, and this rhetoric is often intended to devalue or limit participation from women. Furthermore, the results of a survey I administered to 267 current and former Twitch streamers confirmed that the majority of them have experienced some form of harassment, and that for women, this harassment primarily took the form of uninvited comments about their appearance. Gendered harassment like these are part of the environment of streaming that women must contend with in their work and social lives as streamers. In the next several months, I will begin a series of semi-structured interviews with streamers, viewers, and Twitch employees in order to gain nuanced perspectives on gender, labor, and harassment in live streaming. This summer, I will be working as an intern at Twitch, assisting them in the development of tools and frameworks for addressing problematic behavior on the platform that will hopefully have broader implications on other platforms online. So what my dissertation is trying to do, in part, is to reveal how the work and the play of live streaming is different for women, and to demonstrate how knowing these differences is crucial for mitigating and preventing the spread of harmful behaviors online, as well as understanding the complexity and nuances of life online. I would like to conclude by thanking the Twitch community and the streamers who have participated in my research, past, present, and future. I'd also like to thank my advisors, Dr. Bonnie Ruberg and Dr. Aaron Trammell, as well as the members of the CATS Lab. And I'd also like to thank Dr. Kate Ringland, ARCS Scholar alumna, for her terrific mentorship. And finally, I thank the ARCS Foundation and Twitch Interactive for their generous support of my research. Thank you very much.
Hello, my name is Dan Gardner, and I'm a Ph.D. candidate in informatics and a second-year ARCS scholar. Today, I will be talking a little bit about my research and some of the things I've been able to do the last 2 years because of the support I've received from ARCS. At a high level, I study how different values and systems of authority materialize in technology and media, especially in video games. In my research, I rely on information and computer science, social science, and media studies to examine digital and physical interfaces that mediate various broader cultural concerns. The three primary areas I'm concerned with are security and surveillance, representation and inclusion in games, and accessibility and design constraints. I'll only be talking about representation and accessibility today. Representation across media has been an increasingly important topic in popular and academic discourse the last decade, especially as it pertains to race and gender. In films, criticisms, scandals, and memes related to representation have been regular news. Digital games have similarly struggled with racial and gendered representation since their inception. In my work, I contribute empirical grounding to claims of poor representation in games and work to develop new methods for measuring representation more inclusively, intersectionally, and collaboratively. This chart comes from a paper I published on the largest census of playable characters yet published. It shows the probability of who players may enact in the games in my sample that have a single default character. These are games where players have no choice over who they will play as. Because my data is focused on playable characters, it highlights certain inequalities players may experience taking on these characters. For example, when any feminine player of color sees that they are more likely to take on the role of a masculine green character than someone who looks like their own non-fictional selves, it highlights certain inequalities and trade-offs that some players must accept to participate in gameplay compared to, for example, a masculine white player who is significantly more likely to encounter a character that looks like them. The menus that comprise character selection and customization inherently narrow the field of representation rather than diversify it. Limitations on body types, skin tones, hairstyles, and other physiological and cosmetic features in character creation represent the limits on who games are permitted to be about in a way that isn't as clearly or structurally observable in previous mediums. The other topic I'll discuss today, accessibility, is often about how users or players can or cannot access, use, or play with technology. Or, as it is sometimes put, is a mismatch between the capacities of a user and the features of a technology. In the field of human-computer interaction, we have principles such as universal design, which, depending on who you ask, is either about designing for the most amount of people possible or the most generous average of the population. Or, perhaps more critically, designing for those with the least access or the most mismatch at the far ends of the curve, recognizing that it will only benefit those with more access. What makes accessibility and design constraints so interesting in games is that what constrains designers and what may limit player accessibility are often embodied by the same device, the controllers games are played with. In my work, I am right now finalizing data analysis on a large historical collection of controllers to describe the evolution of controller design over time. This data allows me to examine the normative assumptions about physical capacities of players built into these devices and helps me ask questions about when controllers constrain or can inspire game design. But why study these social implications of interfaces? Well, the primary reason is because they dictate player access and in-game experiences in important ways. As scholar Miguel Sicart describes, in a sort of crass way, we might consider that the only actual activity we participate in digital games is whatever we're doing with our fingers. He writes somewhat tongue-in-cheek, but that we don't feel in games, we press F to feel, to highlight how controllers are an important part of how we connect and interact with games. And more broadly, this highlights the need to investigate other interfaces between us and our digital media. To begin to wrap up, this is just a list of conferences I was able to attend or because of ARCS, all of one of which I was presenting at or supporting presentations on research that I advised. Speaking of advising, another cool thing I got to do was help these amazing women. This is a group of undergrads that I've been advising in the last 2 years all over the country who have already produced 2 publications with another one in review right now at a top-tier journal. I have another team I've been advising for almost a year, but our chance to get a picture got canceled because of COVID-19. They also presented a poster at this year's iConference as well, virtually. I would not have been able to help either of these teams as much as I have without the support I received from ARCS, so ARCS has actually supported them as well indirectly. And finally, while I didn't need a new computer the last couple of years, my daily driver scooter of 10 years finally died last year, and because of ARCS, I was able to replace it almost immediately, which was an enormous help across the board for me in terms of gas money saved and just general access to life. So, thank you again for all of your support, and have a lovely day.
My name is Paul Kurth, and I am a Ph.D. student at UCI, working with Michael France. And today I'm going to tell you a little bit about my research supported by the ARCS Foundation. So one of the big questions that security researchers are interested in answering is what operations are allowed when you try to access a resource. For instance, are you allowed to reach in the cookie jar and take a cookie? My research is focused on the application data that is resident in your computer's memory and how it should be used. Modern processes are composed of several cooperating components. These components all have a uniform ability to access the memory of the application and modify or change its data depending on their operating system permissions. In some sense, this architecture trades performance for security, because while it is very fast, it provides no isolation. In a more ideal world, we'd have a memory layout that looks something more like this, where each component's memory is completely isolated from one another. In practice, this is nearly impossible because components generally share memory resources with one another to do the work that they need to do. This is further compounded when one of the components might be less trustworthy than the others. It might not be malicious, but a bug or vulnerability within that component might allow an attacker to access or modify data it should never have been able to access in the first place. So people have been thinking about this problem for quite a while, and Intel has developed some new hardware called memory protection keys that allows us to very quickly remove access to certain regions of the application memory. So we can effectively build a sandbox around this untrusted component so that whenever it runs, its ability to access unrelated memory is disabled. And its view of the process memory now looks something like this, where it can only access resources it should have been able to touch in the first place. PKRU Safe is our implementation of this idea. It instruments the interface between trusted and untrusted components in the Rust language. It has low runtime overhead, and it can be deployed automatically with only minor changes required by any developers. As we move forward with this research, we're working on ways that we can make the defense more fine-grained and isolate each individual component. I'd like to take this time to thank the ARCS Foundation for its generous funding. It has allowed me to attend several security seminars and symposiums, including the Network and Distributed System Security Symposium, the Usenix Security Symposium, and has also allowed me to purchase a variety of desktop and computing hardware that my research relies upon. Thank you very much, and have a wonderful day.
Hi everybody, my name is Morgan Dragan. I am a third-year Ph.D. candidate in the Department of Biological Chemistry, and today I will be presenting to you my poster on understanding your skin as barrier tissue, especially in the context of these two transcription factors or proteins, OVL1 and OVL2. And so why am I interested in studying the skin? Well, the skin is our very first line of defense against external aggressors, both physical and pathogenic. And if we want to understand how the skin works, we need to understand what it looks like. So on the left image here, this is a cross-section of human skin. And when we're discussing skin barrier, what we're really talking about are those top two layers, the tan and dark pink colors called the epidermis. And so if I zoom in on the epidermis, in normal skin, you can see that the cells within this tissue are very tightly close to one another. However, if you get disrupted skin barrier, these cells are no longer close to one another. And so when this happens, you can actually get inflammation to occur. And so cases where barrier defects are linked with skin inflammation are psoriasis and atopic dermatitis. And what's interesting here is that if you get prolonged inflammation, this can actually transfer to other tissues. So in this case, here's one example where eczema or atopic dermatitis can eventually progress throughout the body, and it can eventually lead to, like, let's say, for example, asthma within the lungs. So one of the major things that my project focuses on is understanding how exactly the skin can communicate with these other tissues, such as like the lungs, or in my case, fat pads. And so, again, as I mentioned before, I'm interested in understanding this in the context of these two proteins and their regulatory proteins called OVL1 and OVL2. And OVL1 is important for, in other words, like the maturation of your skin cells, whereas OVL2 is important for kind of like the growth and development of your skin cells. And what our lab is currently showing is that when you lose OVL1 in the skin, this actually creates a susceptibility to inflammation. And in atopic dermatitis, OVL1 is actually a risk locus for skin disease. And so, as I mentioned before, when you lose OVL1, you can actually get a worsened inflammation or exacerbated inflammation. And so in this case, you can see that the mice have worse psoriasis-like condition, worsened barrier defects, and subsequently worse inflammation. And so one major question, again, I have is, what exactly do OVA1 and OVA2 do in adult skin, and what exactly is their role in terms of maintaining our skin barrier function. And so in order to understand both of these proteins, we've actually deleted both of them in the skin specifically. And when we do this, we can actually see that first barrier defects develop, then excessive nail growth, followed by a separation in body weight. And if you age these mice, all of these symptoms become worse. And so, as I mentioned, here you can see in the graph that these mice have worsened barrier defects. And if you take zoom in images of the skin of these mice, you can tell that the cells are not adhered to one another quite so well. And here is an example of the nails actually being longer in the knockout compared to the control. And so also the next thing that develops is weight loss. And so in younger mice, you can see that they already are losing weight, and as they age, it becomes worse, despite the fact that these mice are moving less and actually eating more, which is contrary to what you would expect. And this weight loss seems to be specific to fat tissues. And so even within the dermal, the fats within the dermis, the skin rather, you can see a reduction in fat content. And so what's interesting here is these mice appear to be burning more carbohydrate stores compared to the control mice. And so overall, what I think is happening is that when you lose OVA1 and OVA2, this causes skin barrier defects. And because of this barrier defects, these mice are now having total body defects or metabolic defects throughout the body. So I'd like to thank my lab and the ARCS funding for all of this research, and thank you so much for your time.
Hi, my name is Gianna, and I'm an MD Ph.D. student in Joan Steffen and Leslie Thompson's lab, and I'm a second-year ARCS scholar. Today I'm going to talk about how ApoE may be degraded by a process called autophagy. ApoE is a lipid-carrying protein, and the predominant lipid-carrying protein in your central nervous system, or your brain. Humans have several different types of ApoE, including ApoE4. ApoE4 is the biggest genetic risk factor for late-onset Alzheimer's disease. There are many hypotheses as to how ApoE4 causes such a great risk. One of them is that ApoE4 levels are lower than the other types. This may be due to increased degradation, although no one knows exactly how ApoE is degraded. Other secreted proteins have been shown to be degraded in an acidic organelle called a lysosome through a process called autophagy. I hypothesize that similarly, ApoE might also be degraded by autophagy. There are several types of autophagy. One is macroautophagy, in which proteins get wrapped up by a membrane, and then that membrane fuses with the lysosome. Another type is called microautophagy, in which the cytosolic proteins enter the lysosome directly. I first tested whether ApoE might be involved in a type of microautophagy requiring a protein called LAMP2A, which has previously been shown to be required for cholesterol trafficking. I knocked down LAMP2A and found that ApoE accumulated in cells, suggesting that its degradation was blocked. I performed a similar experiment, knocking down a protein called ATG7, which is required for macroautophagy, and surprisingly, I didn't see any change in ApoE levels. This suggested that ApoE degradation does not require macroautophagy. However, I also found that when I knocked down ATG7 or another protein, Rubicon, that is required for endocytosis involving autophagy proteins, that ApoE didn't enter cells from the media as effectively. So here I've taken media conditioned with fluorescent ApoE and put it on cells that are not fluorescent. And over time, we can see that fluorescence increases in control cells. However, in ATG7 or Rubicon knockdown cells, there is much less fluorescence, suggesting that ApoE is not able to enter the cell from the outside through endocytosis. In conclusion, we've found that ApoE is degraded by an autophagy process requiring LAMP2A. A future direction that I'm really interested in pursuing is whether this can also happen in animals outside of cell culture. And I've forged a collaboration with a lab at Harvard to try to answer that question. Another finding is that ApoE endocytosis seems to require some autophagy proteins, like ATG7 and Rubicon. I'd like to thank my PIs, Joan Steffen and Leslie Thompson, my undergraduate, Nikki Geller, the entire Thompson Lab, and my many collaborators, including members of the Blurton Jones Lab, fellow ARCS scholar Amanda McQuaid, and collaborating labs at other institutions, including Harvard and in Canada as well. ARCS funding has really helped me sustain these collaborations even during the pandemic shutdown, and that's something that I'm very grateful for. ARCS has also provided me with the support that I need to purchase electronic materials to analyze data, software to analyze data, as well as travel expenses for collaborations and also for conferences. So I'm incredibly thankful for your support, as well as the support of my other funding sources and my departments. Thank you.
Hello, my name is Bree Hoover, and I am a fifth-year PhD student in Angela Fleischman's lab, and I'm excited to share what I've been doing for the past few years. So every day, our bodies produce 200 billion blood cells in a precise balance of blood cell types. The blood-forming stem cells that reside in our bone marrow are the cell types that are in charge of keeping this balance. However, over time, these blood-forming stem cells will acquire a mutation that causes blood cancers. So in our lab, we study a particular type of chronic leukemia called MPN that is caused by the acquisition of this mutation, JAK2V617F, in the blood-forming stem cells. And this drives blood production out of balance and produces too many red blood cells or platelets. And we have a mouse model to study this disease. But what's interesting about when introducing this mutation into mice is that it significantly reduces their lifespan to 3 months of age, where the typical lifespan for a mouse is 2 years. So it's been presumed that this severe drop in lifespan from this mutation is due to a thrombotic event or a blood clot, similar to deep vein thrombosis or other method. What was striking was that when I treated mice with an antioxidant drug called N-acetylcysteine, or NAC, this significantly improved their lifespan. While the standard treatment of care, aspirin for MPN patients, did not improve survival in these mice. Unfortunately, I don't have time to show the data, but you can look for yourself in my paper in Blood Advances from earlier this year, where we found that an interaction between these two mature cell types is what's key for the thrombotic event in these mice. So using a method called flow cytometry, we found that NAC has no effect on platelet activation, and platelets are the cell types that contribute to the blood clotting. However, NAC is blocking this aggregation between platelets and a particular type of white blood cell type called neutrophils. So there's a relatively new phenomena in the neutrophil field called extracellular traps, where neutrophils will expel out their DNA, typically to trap pathogens. But in aberrant situations, like in NPN patients, these neutrophil extracellular traps can contribute to thrombosis. And so this is very exciting data, and this work provides a preclinical rationale for investigating NAC as a clinical trial for MPN patients at UCI. And that's some work that my PI, Angela Fleischman, is working on. So I'd like to thank all the people that helped contribute to this work. I'd especially like to thank Barbara Hemcalo family and the rest of the ARC ladies. Due to your support, I was able to share my work at multiple international conferences, conferences around the country, including in Atlanta and Orlando, and I was able to have multiple resources to thrive in grad school. So I'd like to thank you for your time and attention.
Hello, my name is Jesse Kreger, and I'm a second-year ARCS scholar and fourth-year PhD student in the Department of Mathematics at UCI. I work on the within-host mathematical modeling of disease with Natalia Komarova and Dominique Vaderts. You've probably seen a lot of mathematical models recently that describe the spread of things like the coronavirus from person to person in large populations. I look at the evolutionary dynamics of things like viruses and cancer within a single person that spread from cell to cell. In particular, I look at the evolutionary dynamics of viruses that reproduce, mutate, and are selected for by things like immune responses and drug treatments. These evolutionary dynamics can be easily described by mathematical equations and then cooked into mathematical models. These mathematical models are especially important for viruses such as HIV, which evolve at such rapid rates that they are very difficult to treat. Along with the high mutation rate, there are many other biological processes that affect the high evolutionary potential of HIV. In particular, multiple transmission pathways exist within the body, cells can be infected with more than one copy of the virus, and different viral copies with different viral genomes can actually interact within a single cell. These components of infection can all be described using mathematical models, which then can be used to understand the dynamics of this infection taking place within the body. Since laboratory experiments manipulating each of these different components of infection are difficult, we can instead build mathematical models and simulate them stochastically to replace the experiments. These stochastic simulations can then lead to biological predictions, which give us a better understanding of the evolutionary and physiological nature of diseases like viruses such as HIV and cancer. I'd like to thank the ARCS organization for their generous support of my research, which will lead to better understanding and hopefully better treatment of diseases like HIV. ARCS support has given me access to additional computational power through things like high-performance clusters, as well as the ability to travel all around the world for important conferences. Thanks.
Hello, my name is Alexandra and I'm a second-year ARCS scholar. And actually, I just finished my PhD defense about 2 months ago. So today I wanted to talk to you about what I was working on during my PhD, my main research, and then tell you a little bit about what I'm doing now as well. So during my PhD, I worked in this very highly interdisciplinary field called biomems. And essentially what it is it uses microfabrication and nanotechnology techniques to probe the biological domain. And what I did was use those techniques, use material science, physics, all of these different disciplines to solve different medical and biological problems. The main project that I worked on had to do with stem cell therapy for neural disorders such as Parkinson's, Alzheimer's, and MS, and even depression. And what I did was I developed a material. It was a carbon conductive material, and it was an engineered environment essentially that would be able to not only guide the growth and differentiation of neural stem cells towards specific lineages, but also could act as a simultaneous sensor so that scientists or doctors could know exactly what was growing on that material and whether a certain type of cell was basically produced. And the main thing that I focused on was actually Parkinson's disorder and not only developing stem cells that could turn into dopamine producing neurons, but then detecting the dopamine that they produced at the same time. So it was kind of like a simultaneous sensor and engineered and guided environment for stem cells. So what I'm doing now, ever since finishing my PhD, is actually I've been working at a medical device company, biotech company called Sensor Kinesis, and we've been working on making a portable point of care device, a little bit larger than a smartphone, that can actually detect different viruses that are present from blood samples. And right now, during this crazy time of coronavirus, my company has been working on detection of, not only detection of SARS-CoV-2 virus, but kind of creating a full viral profile of a person's immune response to that virus. And very recently, our company has partnered with Foxconn, which is one of the largest manufacturers in the world. And we're working to manufacture this device and to begin an intensive round of testing. So I'm very excited about that. I'm the head of the microfluidics department in that company. And hopefully we can get something out. We can make wonderful biomedical devices that can not only save lives, but inform treatment. I also wanted to talk a little bit about how important the ARCS program has been for me. Not only did it give me the resources to work on this amazing research that I've been doing, but it really gave me a community to feel a part of and connections for the rest of my life. I wanted to thank everybody at ARCS and everybody who's supporting the scholars and this wonderful program. Now that I'm finished with my PhD, I plan on supporting this program as well. I really think this supporting science, supporting scholars, and supporting this type of research is the best thing we can do for the future of society. Thank you.
Hi ARCS members! Before I get started, I just wanted to say a big word of thanks to you guys for continuing to support us in our scientific research during this very unusual time. I know that you guys are so wonderful to support us in all the ways that you do during ordinary circumstances, but it means even more given the current situation. So with that, let me introduce myself to anybody that I haven't met already, which I think is most of you at this point. My name is Rebecca Riley. I am a theoretical particle physicist here at UCI, and my research primarily focuses around dark matter and other theoretical particles. For example, something called a leptoquark, which I'll be telling you about shortly. But dark matter, I think, is a word that maybe some of you have heard a bit before. But to give you an idea of why dark matter is such a point of fascination for physicists and for ordinary non-scientists alike, dark matter actually makes up 85% of all of the matter in the universe. Now, that is really rather shocking when you consider that there are trillions of galaxies, and within each of those galaxies, billions of stars, that all of us, humans, Earth, everything that we know, all of that is ordinary matter. So all of that, again, those trillions of galaxies, billions of stars, everything that we know makes up only 15% of the matter in the universe. So kind of shocking if you really think about it, that 85% we really know nothing about. We don't know if this type of matter interacts with ordinary matter like ourselves. We don't know how heavy it is. We don't know really where it is, how it's distributed. So these are very important questions that can help us answer how our universe is evolving, what the fate of our universe is, how particle physics works, how we can go beyond what we know already. So very important question. And with my current project, which you'll see on the poster, this particular project, I have pulled in another facet to this dark matter picture, which is the leptoquarks that I mentioned earlier. So leptoquarks are a type of theoretical particle. No one has ever seen them before. Completely hypothetical. You're not behind the curve there. That have properties of both leptons and quarks, which are two different types of matter in the Standard Model that we ordinarily know about. And the question that I've asked is, if we had just ordinary dark matter models with only dark matter, and if we had, in contrast, a model that had dark matter and these hypothetical particles called leptoquarks, could we distinguish between those two possible realities? And so how I'm trying to answer this question, you can see illustrated, and there's a kind of a box in the middle of the poster. So that's the crux of the research. And what we're doing is we're looking out at a neighboring galaxy, Andromeda, and looking for basically what we call annihilation products of dark matter. When dark matter particles get too close to each other, they can actually kind of eat each other, and then that energy where they ate each other, we say it annihilates, they just fracture off into all of these other particles that can then decay. And so what we want to do here on Earth is we have lots of telescopes, different measuring instruments. We are pointing those toward Andromeda, which you can see in the little picture. We point those toward Andromeda, and we try to pick up all of these particle fragments, essentially these decay products. And I'm trying to ask, if I piece these back together, could I distinguish between a dark matter and leptoquark scenario and a just dark matter scenario? And I do that using simulations, scribbling on a chalkboard, using experimental results. It's a very interesting project, and I should have a paper coming out about it shortly. So I'll be excited to share the results with you, perhaps at next year's Scholar Dinner. And I may have some interesting things along another vein that I'm working on, something called a cue ball. It's very exotic, very exciting. So I look forward to being able to share that with you next year in person. Thank you so much again for continuing to support us, and I hope to see you soon. Please stay well. Bye-bye.
Hello everyone, my name is Alex Sutherland, and I'm a first-year ARCS scholar here at UC Irvine. I'm a fourth-year PhD candidate in the math department, where I work with Jesse Wolfson. My general research area is algebraic geometry and algebraic topology. More specifically, I work on solving generic polynomials using geometric and topological approaches. So, what does it mean to solve a generic polynomial? Up at the top in purple, I've provided for you an example of what a polynomial is. We have some variable x, and we're looking at powers of x, possibly multiplied by some coefficients, and then we add them together. When I say that we want to think about generic polynomials, it means that these coefficients, ai, are going to be treated as variables instead of fixed numbers like 2 or 0.01. When I say that we want to solve a generic polynomial, I mean that we want to write down the roots of the polynomial in terms of the coefficients. As a reminder, a root of a polynomial is a number you plug in that when you put it into the polynomial, you get 0. So when n is equal to 2, like in this example, we know how to do this from the quadratic formula, which I've written on the screen and you probably had to memorize when you were in, say, middle school or high school. What we want to do in general is write down a formula like this for all n, not just when n is equal to 2. And moreover, we want these formulas to not just exist, but we want them to be as simple as possible. Now, there's a theorem of Abel that says that we can only write down a solution using square roots, cube roots, fourth roots, functions of that sort, when n is very small, when n is 1, 2, 3, or 4. What's less well known is that Bree gave a solution for the case where n is equal to 5 using 2 square roots, a cube root, and an algebraic function that's now known as a Bree radical. It's like a fifth root, but slightly different. Now, about 100 years later, Klein writes down a solution for the quintic that uses 2 square roots and his icosahedral function. We call it Klein's icosahedral function because he takes the symmetries of the icosahedron and just kind of provides this required function out of thin air. Now, one thing to note is that all of these mth roots, square root, cube root, fourth root, etc., as well as the Bring radical and Klein's icosahedral function, are one variable functions, which means that you only plug in one number to each one of them. My research is on solving the generic polynomial of degree 6, i.e. the case where n is equal to 6. And this builds on work of Klein, Fricke, Hamilton, and others. Now, this case is much harder. There's a conjecture of Hilbert that any solution for this case where n is equal to 6 requires at least one function of two variables. Now, geometrically, this means that we're thinking about objects in two complex dimensions instead of one complex dimension. And that's a lot harder. So how can we make precise the notion of complexity or simplicity here? Well, it's by an invariant that we call resolvent degree. And we can restate Hilbert's conjecture as saying the resolvent degree for solving the n equals 6 case is 2. Now, resolvent degree makes sense across many areas of math. There's a precise way in which solving generic polynomials is universal. And the n equals 6 case is the first situation where we think we have resolvent degree that is equal to 2, i.e. bigger than 1. So better understanding solutions of the sextic in two variables is really fundamental to the theory of resolvent degree. I'd like to thank the ARCS Foundation for your support. With my ARCS funding, I've been able to get a new laptop, new research books and software, and support my research while working from home. It has also facilitated travel to workshops and conferences at UC Riverside and UC San Diego, and support upcoming trips to the University of Chicago and the University of Michigan that I will take when it is safe to do so. Thank you again for supporting me and allowing my research program to thrive.